Alright people, it's late at night and I'm not feeling social. Yet here we are. So, last time we did the sheep brain dissection, I left out the cranial nerves because we'll look at those when we look at the cranial nerves lab. So, olfactory bulbs, optic nerves, all that stuff we'll look at later. Right now we're going to finish looking at the human brain models, and then next week we'll look at the eye and ear lab and the, is that true? Yeah. And soon we will look at the peripheral nervous system lab, which is just really only cranial nerves. So what do we need to be able to identify on human models of the brain? All this stuff that you can look at, like the list of, in your lab manual. And I will show you all of it right now on the model. Everything we see here is cerebrum. So frontal lobes with our precentral gyrus, which is the primary motor cortex. Parietal lobes with the postcentral gyrus, the primary somatosensory cortex. Occipital lobes with the primary visual cortex. Temporal lobes with the primary auditory and primary olfactory cortices. Deep in here we have the insula, which is the primary gustatory cortex. Vestibular cortex is in this temporal lobe as well. Gyri are these raised bumps. Sulci are the little dips. It says corpus callosum, dural septa. Did I bring a good model? I did. Callosum we can really see in the mid-sagittal view of the brain. And it's here, all of this white matter that contains our commissural fibers that connect the two hemispheres to each other. So this is the left hemisphere down here, this is the right hemisphere. Your contralateral control, as you recall, I'm sure. Dural septa. So this brain actually is pretty good. So your dura mater pokes down and makes these septa. The falx cerebri is this region right here. Or of, you look in your book, you'll see it in greater detail. This meninges stretching down in there. And the falx cerebelli is in there. Okay, dural venous sinuses drain your cerebrospinal fluid as it's circulating all around there. Oh, can you see any of those in the models? I'm not going to worry about those in great detail. Let's come back. What else do we need to see? Lateral ventricles. So we can see our lateral ventricles. The ventricle models are easier to see that, but this is the septum pellucidum, this little like membrane that separates the lateral ventricles. So if we pull this off, you can see, oh, there's a ventricle. Be back here, surrounding the thalamus that you see there. And that is full of cerebrospinal fluid to help give the brain buoyancy. Let's go ahead and dig in here so that you can see all that we need to see. So the ventricles, you can kind of see the spaces back here. That's your right lateral ventricle. So your diencephalon is sitting in there, and this fluid-filled space all around there. That's your right lateral ventricle. Behind here, this would be the septum pellucidum, and behind there would be the left lateral ventricle. They're going to drain into the third ventricle that drains into the cerebral aqueduct that drains into the fourth ventricle that drains into the central canal. What else do we need to see? Diencephalon, thalamus, hypothalamus. Infundibulum is this little stalk that attaches your hypothalamus to your pituitary gland. Epithalamus, pineal gland. Whoa, my camera was just like, that was too much, lady. What else? Choroid plexus, you could see on the ventricle model that the professor showed you in class. It was all the stuff that was lacking blood-brain barrier. There's got to be there's probably another model somewhere that has a better infundibulum and pituitary gland. Maybe she didn't bring it home. Oh, that professor. Thalamus, hypothalamus, infundibulum. There's the other half. Something, it's pituitary. It should be there. Whatever. Fourth ventricle, we saw that brain stem. This is the brain stem. Medulla oblongata down here leads into the spinal. Oh, this is the brain stem. Medulla oblongata leads into the spinal cord. Pons, midbrain. It's easier to see on this model. So, midbrain. What do you need to see? Medulla oblongata leading into the spinal cord. Pons, midbrain, corpora, quadra, gemina, cerebral aqueduct. Okay, cerebellum. It's right there. That's not a very interesting arbor vitae. Ooh, that is. Cerebellum, arbor vitae, spinal cord. That leads down into the spinal cord, into the central canal. You saw it in another model in lecture, I'm nearly positive. And then at the end here, these are cranial nerves, but I think because it went with the sheep brain dissection, they're on here, so we'll go ahead and look at them now. We'll look at the sheep brains again later. Olfactory nerves, olfactory tract. So this right here where the eye is your olfactory bulb. And what you'll see in the sheep brain is that theirs is a lot larger. And then this is the optic nerve. Olfactory bulb, olfactory tract. Optic nerve actually comes into here, and then right here at this crossover point that you can't see because our brain's in two. It's the optic chiasm. I'll talk more about that later. And then continuing back from there are the optic tracts. Alright, that's the brain portion of your brain, eye, ear lab. We'll look at the eye and ear portion next week when we go over the special senses.